Good morning, London Calling. Kathy and I are super excited to show you how the NIHR BioResource is using Oxford nanopore technology to transform health research. But first of all, please note the disclaimer from ONT that their product is only for research use only, and also that we have no conflict of interest. So we'll spend the next 15 to 20 minutes first to introduce what is the NIHR BioResource and how we have set up a high-throughput pipeline for long-read sequencing, and then share with you some of our preliminary results as well. So the NIHR BioResource is one of the 4 pillars in the UK genomic landscape. It's all about bringing people together and leading research. But the aim is always for the health benefits as well as innovation. So to date, we have about 350,000 participants on our panel. But our unique selling point is the recallability of our participant, either by genotype or phenotype. So every participant will give us some information about their lifestyle, about their health. They will also give us biological samples. And with that, obviously, we can do genomics. So we have this first stage in the BioResource of banking information. But actually, the aim is always to match those researchers, those participants, for innovation, for health benefit. And the second, maybe, unique selling points of the BioResource is that actually the panel is based on sizable cohorts of participants with health condition, and also those who are, in general, very much underrepresented in health research. So we will have some control, healthy general population, and this is through our collaboration with NHS Blood and Transplant. But we have participants with conditions from bowel, mental health, liver, inflammatory conditions, rare diseases. During COVID, we obviously created a COVID BioResource, making sure that we were doing some good work for this time. And more recently, our activities have actually reached out more to those who are underrepresented. And by these, I mean the young individuals, young people, who are never part of research in general, and also individuals from Black communities. And those are our Decipher and IBO programs as well. So what about long-read sequencing? Well, it all started, like Emma said, for us as well, with rare diseases. When the BioResource wanted to actually make a challenge, it was a challenge, make a difference for rare disease patients. But actually, I will leave you with Kathy, because she will tell you more about this.
Yes. Hello. Nice to speak to you. Thanks for the introduction, Nathalie. So yes, my journey with the BioResource started with this big sequencing project. We were aiming to sequence 8,000 genomes ourselves, and we worked with Genomics England and did 5,000 of their pilot genomes as well. What we really took home after the analysis had finished, we had a Nature paper I've referenced there, but also at least 50 other papers came out on all sorts of aspects of rare diseases and technology, is that we only got a diagnosis for 20% of the patients that were part of the program. Other studies around the world have had similar findings, and we want to know why. We want to do better for these patients, and that's where our journey into to long-read sequencing has started. So our long-read project, it's always we always try and push the boundaries a bit, so we are trying to do up to 22,000 long-read genomes. There are 3 projects involved in this. There's our rare diseases one that started it all, but we've also working with eating disorders, and we have a general population cohort in there as well. It's obviously all Oxford Nanopore. We're using Prometheans, and the lab work is a collaboration between our lab in Cambridge and one in King's College London. All the data is then processed at the University of Cambridge High Performance Computing Facility, and then following our initial publications, this data will be made available through a data access process for the international research community. So the three sub-projects we've got going on, one is related to genes and cognition. It's a dementia-related experiment. We have cognitive profiling data on over 30,000 participants within the bioresource across all those different themes that Nathalie mentioned. All the participants were sent an email and asked, would you like to take part in this app and get your cognitive profile? And we are going to be sequencing 14,000 of those. The majority of those will have 2 time points in that app. And the aim is to look at structural variants and methylation status that might improve understanding of this precognitive decline. We've also got a 4,000 participant eating disorder sub-study, and that's looking at the 2 different extremes of eating disorders. So it'll be about 2,000 people with anorexia, so low BMIs, and 2,000 with binge eating disorders, so really high BMIs. And this is a really underserved population. There are no real drugs to treat these types of diseases, and we're hoping that the research in this area will be really beneficial to these patients. And then finally, the rare disease part of this is 4,000 people where they have had previous genetic sequencing that has not been able to give them a diagnosis, but there will be some cohorts, including family members and some trio analysis going on. And the aim is discovery, but also, where possible, to get research diagnostic reports back into the NHS for verification, to give answers to these families and end their diagnostic odyssey. So it's been quite a journey from piloting these processes in our labs. We have 2 laboratories working on this using 8 Prometheans. So we've got pictures of both of the labs. King's has got 6 Prometheans. You can see them all in a row there. And our Cambridge labs are a little bit more modest with 2 Prometheans. We have both blood and saliva types. You'll be seeing some data on that in a moment. And we have some really quite fresh samples and some legacy samples. So real-world samples, I've heard them described recently. We're aiming for 30x genomes. I'll explain why in a moment. We're doing the variant calling and alignment on the boxes. And with a wash and reload strategy, which means that we can do 24 genomes per Promethean every 72 hours. So between those 8 machines, we can do 384 whole genomes every week, 1,500 every month, and up to about 18,000 every year. So that's how we're aiming to deliver this project. Then we have our bioinformatics team doing all the downstream analyses. So just very quickly, that's the bioinformatic workflow. We've got a lot of work going on those Promethean towers that Gordon was showing, how many calculations they can do a second. Well, they're doing a lot of the hard work with the base calling and alignment, but then the work continues at our high-performance cluster at the University of Cambridge, looking at the 5 main variant types and doing all the extra pipeline work there. Beyond that, we also do, we're setting up a lot of QC analysis to make sure that the participants we think we're sequencing are the participants that actually got sequenced, that there have been no mistakes in the lab. We're also looking at cross-contamination between samples, or making sure that there isn't any of that. So there's sex checks, we check the phenotypes, heterozygosity, we're looking at the ancestry, which factors into the downstream analyses, but we're also looking at the quality of the sequence that we generate, so looking at missingness of data, Hardy-Weinberg, allele frequencies, things like that. So I've just put a quick picture in there. I always like a PCA, so this is the ancestry analysis. The coloured dots are all from the UK Biobank GWAS, and then the black dots are our first 1,500 people, and you can see they overlay really nicely onto that analysis, even though they are different data types. So we are about 6,000 genomes in, but because the bioinformatics takes a little bit longer and we had to get our slides ready for London calling, this is a snapshot of the first 1,500. They're spread over the 3 different cohorts I mentioned. You can see that our cohorts, because they are UK cohorts, they are tending to be very Caucasian biased. We deliberately skewed some of these early samples in the genes and cognition cohort to be some of our more diverse genomes in the bioresource. So that skew will go away as we fill in the 14,000 samples there. But you can see that we've got a good range of ethnicities and genders, and you can also see the eating disorders cohort is actually heavily female skewed. Even in that first small subset of people we've started, we found one person XXY in there as well. The other complication we have with these real-world samples is we use both blood and saliva samples. So any plots coming up, if the plots are in blue, that means there are saliva samples, and if they're in a pinky red, there are blood samples. We also, because these have been banked over 15 years, a variety of different extraction methodologies have been used when generating these DNAs. I'm not saying anything about you should use one technique over another. These are just what we happen to have to use. The DNA is being extracted, we have to cope with those, and we do see differences in the method or because of the age of the samples. So that's just a quick spread of the different methods we've used. Here's a first look at the data. So the N50s for blood do tend to be a little bit higher than the N50s we get for the saliva samples. They are a little bit more degraded and harder to work with. The depth of coverage, we are aiming for 30x genomes in all the cases. It's a little harder to manage with some of the DNA inputs, particularly the saliva ones, so there is a broad range there. And the Q scores generally all high 18s or 19s and above, so nice Q scores coming out of this analysis. This is the problem with working with saliva. It won't be a surprise to you. Blood, of course, lovely to work with, pretty much every read mapping to the human genome, but the saliva samples obviously include microbes and sometimes people's lunch. So some of those samples are really good. They do map really well to the human genome, and others you can see less than 50% of them are mapping. So we do have to over-sequence some of these saliva samples to get to the 30x coverage. So they are a lot more challenging to work with. We did do some benchmarking with some of the genome in a bottle samples. So this is a slide summarizing 6 samples we ran, 3 in Cambridge, 3 in London lab, to make sure that the labs were producing comparable data. And you can see that the F1, the precision and the recall are all working out really nicely when we run the samples. The reason we want 30x coverage is you can see that the SNV plot there is starting to plateau at around 26, 27x. So we think we're calling all the variation in the SNPs there by 27x. At similar depth of coverage, it's about 0.95 for the structural variant. So the more coverage we get, the better it is for the structural variants, but we have to work with the samples that we've got, and it's just not always possible to get above 30x. Looking at the count distributions across the different variant types, so we've been looking at all the types, structural variants, SNVs, indels, deletions, and the methylation there. You can see that across all those 5 sample types that we've been trying to optimize, the numbers come out pretty similar. A little bit less in some of the cases for the saliva samples, but as we've explained, the coverage is a little bit harder to get for all the saliva samples. And you can see there's another slide coming up that the skew in the Quiogen samples is because of those non-Caucasian samples that we've deliberately put in there. So we were quite pleased to see the consistency across all the variant types with our data set. One interesting and pretty recent observation, so we're still looking into this, is on the methylation. And we've been finding that the pass rates do vary a little bit in the saliva data set. They seem more consistent in the blood data set. So there is a correlation between the pass rate and the coverage in our saliva samples that may well relate to this lower confidence in call rate when we have a lower coverage. So it could be that we need to do as best we can with the coverage to get the best methylation data out of our saliva samples, but we'll keep looking into that. This is the picture with the ethnicities, so it's no surprise that the SNPs and structural variants are more prevalent in our African and Middle Eastern populations within the bioresource, but the methylation signals are pretty consistent across all the populations. And then the other thing we wanted to do was prove that the technology was working as well as the previous technology we'd used. So as we said, we'd done a lot of work on rare diseases. So we went back to some of our rare diseases where we had confirmed cases and fed them back to the NHS and had them validated by the NHS. And we wanted to make sure that this technology would pick up all of those variants as well as all the other extras it adds. So I've got a couple of slides coming up just looking at a SNP, a de novo SNP in a trio. It's an example from one of our pulmonary arterial hypertension rare disease cohorts. Got two examples of compound heterozygotes. They both happen to be in our primary immune disorders rare disease cohort. And it's really nice seeing this in the nanopore data, how compound hets can work, and that does not need to have a trio to determine this. And we've also got some examples of some structural variants that we found in our retinitis pigmentosa, or one of our eye disorder cohorts, in our bleeding and platelet disorders cohort, and another one in pulmonary arterial hypertension. So the first one's fairly standard. It's at the top of the graphs, you're looking at the original short read data, and then the bottom of the graphs is the trios with the long read data, and usually with phasing turned on. So if this was a SNP, it's only present in the proband. That's why it's a de novo cause. So that was simple. The next ones are compound hets. In these cases, the two variants were in genes either 8,000 bases apart, or one of them is 25,000 bases apart. And so this shows the power of long-read sequencing to have those long fragments so that you can phase the genomes. So in this example, you've got the whole trio. One of the variants comes from the mother, one comes from the father, and in the proband you can see that one chromosome has the mother's variant, the other chromosome has the father's variant, and that means both chromosomes, both copies of the gene have been knocked out, basically, which is why the person has the phenotype. And normally with short read sequencing, you need to sequence the whole trio to understand that. With long read, we could get away now with just sequencing the proband. And this is often really important to us because we don't always have the parents available or other family members in order to do that. So being able to do the phasing is really exciting, and I know our primary immune disorders cohort very excited to look at more of these. So this one's just another example. In this case, we had a mother and a brother rather than a father to do the phasing with. And then the final ones are some deletions. So this one's a large homozygous deletion, so you can see the breakpoints come out really, really nicely there, and that both chromosomes are basically taken out by this deletion, which causes the phenotype. This one was a deletion and an SNV, so you should have zoomed in a little bit more, but there is a heterozygous deletion in there that matches what we found previously. This one is another large deletion that takes out the 5' end of the gene, also explaining the phenotype. And then finally, the other thing we're really interested in is methylation. That's another really powerful aspect of this technology. So this is an example from one of the genome in a bottle samples looking at PEG3, which is well known for imprinting. And you can see in that bottom trace there how the lines diverge, and that one haplotype is heavily methylated and one haplotype is not methylated. And this is exactly the sort of signal that we're hoping to see in other genes throughout the cohorts that we're looking for, and hopefully explaining some other phenotypes. So, in summary, here at BioResource, we're building this resource of up to 22,000 human genomes, all based on the UK population. We've demonstrated that we have a workflow that can scale to 48 human genomes per week per Promethean, with 30x coverage. We've shown you an example of the sort of data quality that we're obtaining, both using diverse sample types, so that's blood and saliva, old and new samples, and also different participant demographics. And we've demonstrated that we confirm all the diagnostic type variants and methylation signals that we would expect to see for the analysis that we want to do moving forward. So in summary, BioResource is actively enhancing multiple health themes with rich genomics data sets, and we're hoping to revolutionize gene discovery and diagnosis rates and treatments and clinical care for patients all around the world. So thank you for listening. There's some photos of the lab team, the bioinformatics team, who are also really key to this. We're a bit too shy to give me pictures. thank you to all of them, to all of the BioResource, without whom we wouldn't be able to do this work, our funders, and of course all the BioResource participants. And if you want to know more about us, we've got emails, we've got Twitter, or X as it's called now, LinkedIn, we've got a web page. Do get in touch with us. Thank you very much for listening, and I think we're open for questions now.